we have already seen that this conventional drug delivery, which have got limitations of this, and we have gone to increase the efficacy we have done. So most of the times the targeting method we are having a active and the passive we have discussed. In the passive transport we are concentrating on the enhanced permeability and retention, that is EPR effect. In the active transport we are taking one carrier help, and we are taking some approach that is specificity is main in that. In the passive it is not having a specificity, we have already seen that. So in order to enhance more passive effect, we need to make the reticular endothelial system, which is should be shut down by making the molecule more hydrophilic. One. The second one is by making the reticular endothelial system busy with the dummy microspheres or nanoparticles. So by injecting them in advance and making them busy, and then we are going to give to enhance the EPR effect in the passive targeting. In the active targeting, we are using a specific moieties, chemical structures that is having a conjugation with a specific receptors that is going to take to the particular organ or the site or the cell only, so that it is increasing its concentration. We have already discussed. In the active targeting, it can also be achieved by using the magnetoliposomes, where we are using the external magnetic field to make the concentration of iron-based nanoparticles in a particular organ. So this is another approach, and we have already seen that there is a lot of disadvantages in the patients which they are operated in the heart with the pacemakers, or somebody is having some of the plates in the orthopedic surgery. So in those cases, these magnetic field effect, they cause the deformities in the existing structures which are inserted as implants. So such things we need to take care here. And we have seen in the third terminology is the inverse targeting. So it is by the suppressing the passive targeting by giving the dummy capsules. We are naming it as the inverse targeting, so that our passive targeting will be more having a more EPR effect. So that is enhancing the reticuloendothelial system busy. This is actually what the inverse targeting is. Most of the times you may be getting confused with so much of terminology. They are going to get entering into different levels. So if you understand the concept an individual and taking that into the particular, fitting into the particular scenario or a principle, that you will be able to understand. First, we will be learning these approaches so that later onwards we can directly jump into that word without any problem and misunderstanding. Most of the times we have seen that the targeting to the cancer cell, there are two major approaches which is pertaining to the cancer, and we are going to have, this is the one is the pH response, another one is the redox potential. These two are the more having variations when compared to normal in the cancer cell, and we are seeing this as an approach. And another one what we have discussed in the previous class is delivery to the brain, where the delivery system to the brain in the process of changing therapeutically active molecule from the across the brain, treating the brain diseases. For that purpose, we have got so many challenges like tight junctions, lack of pores in the brain, that is blood-brain barrier, so that is causing a barrier effect. So we have got a different approaches, invasive and non-invasive techniques to enter into the brain. And the methods of delivery, we have seen that we are using the lipid moiety by enhancing the prodrug concept, and we are also having a disruption of the blood-brain barrier by giving a hypertonic mannitol solution to make the disruption, so that we will be having enhancing permeation. So these things which we have already seen as an alternative to the direct injection to the brain at the lumbar pace by the intrathecal injection or intraventricular injections. So we have seen these approaches of as an examples, and we have discussed earlier. So these were a carrier-mediated transport is the one which is already existing in the blood-brain barrier. We use the same transport for our drugs also by mimicking the ligands which are having a similar affinity to the receptors, then we are going to enter into the brain easily. This one also we have seen. Disruption of the blood-brain barrier by using inert solution, hypertonic solution of the mannitol and arabinose is one methodology, so that we are entering it. This is another approach. Now we are having different delivery systems. We are going to deal, and the first one more preferably which is being used is nanoparticle. And there are two strategies are applicable in the drug targeting by using a nanoparticle is a physical and chemical targeting. And this is also we have seen. The physical targeting is based on the size, shape, charge, and the stiffness. That is the flexibility of the particle. These parameters that influence the tissue accumulation, adhesion, and cell uptake. This is actually what is present in the physical targeting. The chemical targeting is the employ the molecular recognition, just like your receptor ligand concept. So to decorate the surface of the particles, so molecular units response to the disease, that is the diseased environment or the remote stimuli, they will be specifically attached there because of this chemical stimuli or response. Then we will be making a targeting possible. So this is where we have stopped in the previous class in the ligands and antibodies are the major thing which are specifically targeting into the particular receptors that we can give by conjugating the dummy nanoparticles. They are loaded with some drug or drug loaded matrix systems of the nanoparticle or drug loaded nanocapsules, which we are going to give and by conjugating with the antibodies. These antibodies are having a specific recognition to a receptors which are present in the particular cell. So we are going to enter in that particular vicinity specifically because of this antibody and receptor, that is antigen combination that is taking to a drug targeting. So this one which we have already seen. Now I would like to make it little bit enhancing your concept, and we are going to elaborate it here is now of the targeting cytogen nanoparticle, which has been already been done in the literature, which have been done research to the brain using a mouse monoclonal antibody has been studied in long back to see that that is given as a OX26 and against the rat transferrin receptor. So how it is going to have a targeting in the particular one? So the transferrin receptor has been reported to be high abundance at the blood microvascular endothelium. That is the reason if you give the monoclonal antibody to that particular receptor, we are going to have more abundance of the drug. So the goal was to inhibit the caspase-3 like a protease activity using the peptide that is being given. So most of the times we will be giving as a course initially. It is given in a chemical structure N-benzoyl oxycarbonyl DVD fluoromethyl ketone peptide because of its therapeutic significance in the treatment of the stroke and the related neurological disorders which are going to cause by the abnormalities in the brain. So this has been came into the existence that we can use these nanoparticles loaded with these particular enzymes to treat the diseases. So another strategy involved the use of the biotin labeled PEG to coat the cytogen nanoparticles and the streptovidin labeled antibodies for the conjugation. So this OS26, that is the monoclonal antibody conjugated with the nanoparticle can penetrate into the brain, whereas the OS63 that is not containing this monoclonal antibody, they cannot penetrate. It means that the blood-brain barrier has got some transferrin receptors. If you make some attempt by giving a antibody to that particular receptor, and that is going to have a more targeting and increasing the concentration and penetration power. So this is actually resulting into a fifth unit, which you are going to enter a little bit later in the blood-brain targeting, and you where you are going to see your delivery system and the gene therapy and nucleotide delivery and these approaches and pharmacokinetics we are having in advance in the later stage. Whereas the targeting to the cancer, it means we have found out one carrier that is good to that we are going to have utilize it without causing a damage, without having a disruption, we are giving without having a puncture or inserting a needle. So similarly, if you look at the targeting for the cancer, one approach which has been came into the existence long back, is epidermal growth factor receptor. If you see here, everything is a receptor. What is this receptor? This receptor is nothing but a protein. Where the protein is coming? This protein is coming from the single cell. What it is present as a zygote in the mother's womb in the starting day, it is already been made available in that particular template present there itself. But it is triggered to produce this particular protein in the particular time and a particular place and according to the need. So this is happening what in the cell is growing into a body, human being. So this is always, it is already present in the gene, it is already got encoded to have all these things which is being required to be done in a particular age, at a particular time, so in a particular place. So this receptor is highly expressed in the breast cancer. It has been found. Nowadays everybody is finding, and this is the biggest problem, and has been used with the PLG and nanoparticle containing rapamycin, that is the drug to treat the particular cancer, that were surface conjugated with EGFR antibody. If you give this rapamycin directly without conjugation, it is not having a targeting effect. Whereas if it is causing with having this antibody of this receptor, then it is having a high specificity there. It means my body entire thing is good and doing good to me. I am protecting it. Only the damage which is coming is in a particular part only, that is rich in certain receptors. I prepare an antibody for that, and that antibody will particularly see it in that only. Like I have got so many locks in my house. Each lock has got its own key. I cannot open another lock with another key. I need to use that key only for that particular lock. In the particular phenomena, I can have a specificity to insert and open it by having that particular one, otherwise the remaining thing will be safe. So the specificity of structural, chemical structure, which is present at the molecular level, we are using to have this particular effect of getting a targeting. So this antibody conjugated nanoparticle can also be used for the targeting delivery of the nucleic acids, that is going to have the later. The use of siRNA, that is a single interfering RNA as the therapeutic agent, that is a small RNA, that is a small strand, that small interfering RNA, and it is a therapeutic, has not a lot of promise, but main obstacle is to prevent it from the degrading upon the drug delivery and improve targeting. So this is actually a problem. It is the hydrolysis cleavages will be present. So that is the reason there is a CALLA01, that is a coded form of a nanoparticle which has been used to deliver this particular small interfering RNA that will decrease the cell growth. And it is the first nanoparticle of its type in the entire human trials which has got a success. It made that as I can do something by the formulation manufacture, not by the inventing the drug. I am coming to the formulation manufacture. There are two we have got in our pharmacy. In a product oriented B pharmacy, you are studying it, B pharmacy course, there are got two things which are present in the product orientation. One is inventing a new drug, inventing a new formulation. In both the cases we are working. Our M pharmacy is mainly targeting with and dealing with the invention of a new formulation. There is another aspect which you have got a product oriented to the service oriented. Service oriented means doing a service at the hospital. That is done by the pharmacy students. That is not our job. So they are providing the service and giving information and giving counseling to the patients and doctors and nurses and lab technicians. Everything is going on with their service. Now we are not dealing with the invention of a new drug, but we are dealing with the invention of a new disease, a new problem solved by the new formulation to treat the disease. So this is actually what this approach first which is been done. So the nanoparticle formulation could be able to deliver particularly to the tumor cell. Otherwise, this siRNA will not function. It will be cleaved. It will be cleaved. So this is actually what our formulation beauty is. We feel proud that we are in the similar business and we are doing the same thing and learning it. Now the question comes, whenever you are doing, why this particular one is happening in the body? Wherever you know that the cancer is a cell growth. It is unstoppable cell growth. It is keep on doing. Why it is happening? That is the biggest question whenever you are asking the question in the treatment of the cancer. So I'm going to give you some of the nomenclatures so that we will be able to understand and recapitulate the previous learning, what we have done in the previously in our B Pharmacy level, and then coming back, bringing them back. So what is happening in the cell, cancer cell? The unstoppable, there is no stoppage of the cell growth. It is keep on growing and growing. That extraordinary growth when compared to a regular cell, it is causing a requirement of more nutrients. What is more nutrient which is requiring? Because it is multiplying it requires for the multiplication its proteins and cellular material. For that purpose, it requires all the raw material. For that, it will take from the body only. When it is being supplied to the particular tumor, which is not good to us and it is not required for the multiplication, and the remaining cell will be deprived, and it is having a depletion of food nutrients. That is one problem. And we are going to have multiple breakage and converting of cells into multiples of so many, and we are getting a tumor that is causing a pain and protrusion, and it is causing the damage to our body by blocking the blood vessels, or it may be hitting to certain particular part, specifically in the brain. If the tumor is becoming bigger and it is giving a pressure, it is going to cause to a particular part of our brain. That particular part which is dealing, you will be getting paralyzed. Somebody may not sense the smell, somebody may not sense the hearing, somebody may not have the walking ability, depending upon where the tumor is touching the place where it is present. Most of the times you may not be remembering. So some of the cancer patients which are in the brain tumors and the cancer cell which is present in the brain, they will not be able to see it and they will not be able to sense it. Only the thing with the mechanism which is being done, that suddenly, all of suddenly, they lose their sense of taste, and they will be getting confused. Why I am not able to sense the taste of sugar or a salt? Both are appearing same to me. Then it is getting tested. Then it will be known that yes, there is a problem in the brain, and the brain tumor is getting bigger, and it is touching to the area where it has got ability of the sense perception of a smell. So this is actually what the cancer cell is causing a damage by multiplying it. So why it is happening? We have already seen that each and everything in this particular one, it has got a birth, some work to do, and after that it will die. But in the case of cancer, that is not happening. That death is not happening. So there is inbuilt technology in our body is called as apoptosis. This apoptosis is a self-death. And the body will make the cell to die by its own, so that we will be generating a new in the place of the old one, and the old cell will get shrinked and it will be getting disappeared. It means, say for example, you have got a life of your RBC cell is 120 days. So after 120 days, the RBC cell which has got its own trigger mechanism to get shrinked, and it is studying its all cell organelles, and it is taken by the phagocytosis, and it is recycled in the body, and it will vanish. In the place, again, some more cells will be generated. Imagine the situation, some more cells are getting generated, and old cells are not dying, and how can the body will cope up? And you cannot even feed all the cells regularly, whatever the food you are going to take it is not possible to survive. That is what is happening. It means the body has got intrinsic mechanism to make the cells to die, and when they are not dying and they are causing the damage to us. This process of killing the self-death and making a suicidal of the cell, which is already making it to die after a particular period of the time, which is already been pre-programmed, if it is happening correctly, then it will not result into a cancer. That's cell cleavage or the cell death is not taking place, it will be resulting into a cancer. Now we are going to talk to that particular level so that we will be able to understand why we need to treat the cancer and what is happening behind before the cancer is appearing in our body. The term is called as apoptosis. Apoptosis is a form of programmed cell death that occurs in the multicellular organisms most of the times, and in the unicellular organisms, and you are going to have that cell is getting killed or engulfed and it is died and it is got. death. And in the multicellular organisms, this should happen. This should happen. Otherwise, it will be becoming like a big hill. The body will become big hill and keep on feeding it. Otherwise, it will not survive, because if you keep on going, there is no cell death. So the biochemical events lead to characteristic cell changes and the death that will be already programmed, and it will happen after a particular period of the time without our intervention. It is in the subconscious way it will be happening. What is the subconscious way? I'm telling you and speaking to you. I'm breathing without my consciousness, and consciously I'm not breathing. Automatically I'm breathing, and my this is happening. In the similar manner, how I am breathing, the same phenomena is happening in my body, like apoptosis. These chemical cell events which are present, it means there are so many chemical reactions are present in our chemical laboratory of our body. There are lakhs and millions and trillions of chemical reactions going. Similarly, trillions of the chemical reactions are synthesizing a new moieties. So trillions and millions of the chemical reactions are getting diagnosed, and it are getting sensed to take the changes and cope up with the changes. It means the synthesis, analysis, and reactions, progress, everything is going in the plant if you are considering our body. So these are all the pre-programmed conditions which has already been praised, and it is present already in the DNA structure of our body, which is present in a single cell. It is already been coded. So at the particular point of time, that particular gene will trigger to liberate some of the extraneous materials or the chemicals that will cause these chemical changes and events that will characteristic make that particular changes and make the cell die. The changes include the blubbing and the cell shrinkage and nuclear fragmentation and chromatin condensation, and you are going to have the chromosomal DNA fragmentation and the global mRNA decay, and the cell will lose its function, and it will be getting smaller and smaller and day by day, and it is not having any function and considered by the body as a fragments, and they will be removed by the phagocytic mechanisms, and they will be taking and recycling them, and to provide a food to the new cell which is being generated. So this is actually what the concept of apoptosis, which is helping us, so that we are rejuvenating it. I am becoming a different body and a different person every day. It means a second before a Jagadishi is not same as that of now the Jagadishi. There are so many chemical changes which have happened, and so many microorganisms and cell died within the body, and new has emerged. It means the present body is a new than yesterday's or previous seconds. It is a different with respect to our physical, with respect to mental, with respect to our maybe a spiritual, with respect to our knowledge, wisdom. So a second before I may not have the wisdom or the knowledge, maybe I am carrying, now I am making myself a different person. So this is where our education is providing to make a person from a state of that to higher level state. It means we are calling in Telugu as a unna sthiti nundi unnata sthiti. It means we are staying from an original state of one to a higher level. So this requires this apoptosis process. This rejuvenation is required. Otherwise, we will not be having a same. We will not be like a same thing which is prevalent for years and years. There will not be any change. This is inevitable. It requires to be present. What is this mechanism of apoptosis? Apoptosis is mediated by the proteolytic enzymes called as capsages. Previously, we have seen the capsages are different named 01, 02, 03 like that, which trigger the cell death by cleaving specific proteins in the cytoplasm and the nucleus. These capsages exist in all the cells, but they are inactive. They are inactive precursors. It means just like a prodrugs, and they will not act. Are the procapsages. They will be existing, but they won't work. They will work only after a particular period of the time. They get a chemical trigger. Then the trigger converts the procapsages into the capsages, and which are usually activated by the cleavage by another capsages, which are present in response to the other body, and producing a proteolytic capsase cascade. It will cause the cell death, and it will cause the rejuvenation. So it means this body cleaning process is always existing in our body. So if it is getting interrupted and apoptosis phenomena is lost, it can be due to Procapsages are not converted into capsages, and capsages are not liberated and acting in time, or they are got blocked by another chemical or the drug which we have already given. This is what happening. We are giving a drug which is having a problem in a treatment in a particular part, which may be having a part of a problem in another way, in another protein. So it may be getting blocked. When it is getting blocked, it won't work. When it is not going to work, that phenomena will not be stopped. When it is not going to be stopped, it will be progressing. It causes the disease. This is actually what they called as chemical way of triggering and making a reaction, that is adverse drug reaction or a drug-induced diseases. Most of the times we are finding our laboratory, we have got a latrine cleaners, so that is whatever you use, the acids and the harpic, everything, there are plenty of carcinogens which are present. The benzene solvent which we are using, it is proven to be carcinogenic. And we have got so much of cigarette smokes which are present in hundreds of materials which are going to trigger the even a passive smoker also carcinogenic tendencies. We have got so many ill thought process. We have got so many 6,000 odd thoughts which are going to be progressing continuously in our brain, and negative thoughts which may interrupt our apoptosis. So making our ego, it means that I feeling. It means it will not allow to give anything. It want to retain. The body want to retain. It won't want to kill. It won't want to give anything. So in this particular case also, that mentality triggered problems in the disturbances of the apoptosis may occur. In that case, it is also a trigger point, like just like external physical triggers, a mental triggers, and it is known that there is a spiritual triggers. My religion and my thing is only the correct. I am only right, and my God is only existing. Others are not existing. This is actually another trigger. So in the societal, my caste or my creed is only the best, and the other person should not live. So I need to protect. I need to stay only. So it will not allow the apoptosis. You are triggering your brain not to do the cell death. You keep yourself. The person will get a cancer. It means the ego or the mechanism of your chemical and the biological and your subtle mental abilities which are going to cause the thought process is also having a trigger. So if you are feeling, say for example, if you have got a good food which is being present, you have ate a long back, and the moment you think that particular about that food, you will get a watering in the mouth. What is that giving? That food is not available in the body. We need to consider that also. We should know about it. At least we need to correct ourselves. We need not correct the society or anything, so that we will be having a better. So that this is actually the worst phenomena which is causing the cancer and making us the trouble and preventing the cell death and causing its presence and more and causing the cancer. Now we are entering into the specificity to the targeting for the vaccine. A vaccine which is the phenomena which is having a different modes. We have got a dead vaccine, which is a death after killing the bacteria or virus. We are taking like a Covaxin is a dead virus. Maybe we are using mRNA and making a protein synthesis and triggering the similar antibody. The protein which is created, which is going to trigger the antibodies in the body, whatever it may be, it is required to be acted in a particular place. It should not be attacking everywhere. It is required to be acted. So we need to target vaccines also as in a delivery system. We know that we are giving a vaccine in the intramuscular injection. Sometimes we are doing a intradermal. Sometimes we are in intranasal. Sometimes we are giving as a oral. In all the cases, we are having a specific site of administration, and we are going to find out. So for that purpose, we have got an approach like a dendrimers. So these dendrimers are having a specific approaches to carry these vaccine cells, which are going to have a specificity because of their branched in nature to hold these vaccine moieties in their branches and taking to a particular level depending upon the extent of branching. So this has been proved and established that we can use it a good system as a one nanoparticle nanotechnology to deliver this vaccine. So the dendritic cells are thought to be most potent antigen presenting cells for the stimulation of the naive T cells, but are difficult to target because their low number compared with the macrophages. So for the vaccines, an ideal target molecules has been endocytes mediated receptor, that is TEC205. It is also called as a CD205 because of its specificity in the dendritic cells and its ability to provide the receptor and mediate endocytosis to the particular vaccine. So the cross-linking densities must also be considered during our formulation manufacture in these cases whenever we are using for the vaccines, because this is a burning topic now. So if you want to deliver a vaccine, so we have got so many precursors and excipients which are having, and also adjuvants. In the adjuvants means they don't have any antigen inducing or antigen capability by themselves alone. But when you are mixed with these antigens, they will trigger and in a symbiotic manner. In those cases, we need to add them, and this this delivery system, which is having a different cross-linking densities, must be considered, so that to what extent I need to stop this dendrimer particle to carry my antigen to get a better effect when constructing the conjugated nanoparticle, as they may affect the targeting of these dendritic cells and its activation. Most of the times we use a different stimuli responses as strategies. I hope you understand the complicated topic. I would like to repeat if you are not able to understand also, we will take a little bit more time also, but it is very much crucial at the molecular level to understand what is going on, how the different triggers is changing my biological responses within the cell at the molecular level, so that I need to treat them. Otherwise, I cannot treat. I cannot give the formulation to them. So this is what we are going to track and attack, so that I am having a specificity very high. So this is actually what is called as a stimuli responsive strategies. So there are different stimuli that will help us to take our drug specific to that particular site. So one response, that is a stimuli response, is pH. So the target is acidic compartment found in the pathological tissues, most of the times in the tumor sites, because they are having a cell growth requirement of a glucose is very high and oxygen, and it is causing cleavaging of this and getting the pyruvate process more and glycolysis and causing the increasing the acidic chemical generation and decreasing the pH. So acidic compartment within the cell are in the endozones, so that will actually acting as a target to us, that is having a deviation of our regular pH. So this is actually one target. Another one is the thermal. So responding to the heat, the delivery system will work. Now you may get the question, sir, how can it be possible whenever I am having it heat, because my body temperature is 37 throughout my body, it is present at 37 only. But sometimes what will happen, if your body is having overflow in a particular part, or your body is having a capillary, it is constructed, or in a particular inflamed tissue where you have got a swelling, you have got a little bit more heat, localized heat there. So that localized internal heat or external heat applied. So heat applied from the external or from the physiological changes, that will act as a target to us and taking a moiety there by making a dermal responses changes. This is one. Another one is the light. Most of the times this can be used externally applied light, so it is actively guiding it. Most of the times it will be used very much in the detection and diagnosis, because most of the we have got a fluorescence and phosphorescence. So we have got that fireflies. If you see that they will scintillate the light during the night, you will find it by the external application light, and they will be having a phosphorescence. So it means if you are giving external light to that particular part where that particular moiety which is having a phosphorescence is present, and we are tagging them in the nanoparticle, then we will be able to set this is actually called as luminescence, and it will able to detect inside the body part when they are traveling. It says that we will be able to see whether their targeting is going or not. This is another excellent thing to measure the targeting index, whether it is properly going or not. Another response which we can have the stimulative response is redox. So you may be having a questions in your exam It may be a lower marks in the sessionals and higher marks in the exam and bigger questions. Sometimes they will be asked to list, sometimes they will be asking elaborate, sometimes they will ask to explain and describe in the deeper level. So this is the lower level when you need to just name that one, and we are going to the individual one, you may get the 10 marks question also. pH responsive systems and dermal responsive systems, like that you have got individual. So that we will be having that stimuli and adopting a system, and we are going to have a deeper their understanding. So the redox system is a response which is going to the target in the region where the reactive oxidative and reductive species are abundant, like in the inflammation and the tumor sites. So there we are going to target there. Another one is enzyme response to the specific enzyme which is liberated in the regions of the high protease expression, such as in the cancer tissues. So this is actually what is present more there, and we can attack that particular one when compared to a normal cell. So where there is a deviation, where the apoptosis is not going on, where if you want to have the sense of changing in the response, if you are able to know it, so accordingly we will make a device, or it may be a diagnostic kit, it may be a formulation which can use to have that particular part attacked and targeted. And specifically, that is the reason we are having a tremendous advantages. Because of that reason, we are learning all of them in the classroom and applying, if possible, in our project in the next year. And learning extension in the Google or doing literature survey in the interesting topics, and making understanding of a particular points in writing in a paper, and going further to learn and clarify that particular doubts, whatever which is present in between, because it is vast. It is keep on changing. Nobody can making a perfect in this particular journey, because it is keep on changing every day. So what we have got in the previous year, by this time when the COVID is going